Hello, everybody. Welcome to our monthly VanBug Bioinformatics seminar. My name is Emily. I'll be your host for today. Oh. We're good to start, right? Yeah. Okay. Before we begin, we'd like to remind everybody on Zoom and in person to please mute your mics. All of your tables have microphones as well. It'll show like a flashing light when it's your turn to speak. So just to ensure that none of the speakers are interrupted mid-sentence, please make sure those lights are off until our question and answer period, or if the speakers will be accepting questions mid-presentation. Just a note for you guys, this event is being recorded. We are going to take a video recording of this event, which we will likely upload to our YouTube page for anybody who was unable to attend our seminar in person today. If you have any questions about the protection of your personal information, please contact William Xiao. His contact information is on this slide. For our schedule today, we are currently in the announcements portion, and then we will have our introductory trainee topic from Nicole Nutza, and then we will have our featured talk from Dr. Inanc Birol. Hopefully we'll end on time by 7:20 because we are going to have a little bit of a social mixer with some pizza and drinks, so please stay tuned for that so you guys can all chit chat and mingle with each other. Hello, welcome. I'd like to thank our wonderful VanBug organizing committee, especially our faculty members, Dr. William Xiao, Dr. Amy Lee, and Dr. Faraz Hotch. Thank you for all of your support and guidance in helping these events run smoothly and hopefully well. I hope you guys enjoy. As well, thanks to all of our volunteers for helping us out. So for our upcoming VanBug events, our next one is on November 20th, but they'll be going monthly all the way up until March of next year. We are releasing details about the events on our website, vanbug.org, so you can keep up with that website. Alternatively, instead of checking that website all the time, you can join our mailing list. So if you go on our website under the contact page, you can fill out this link. It's to a form, and you'll get emails about all of our upcoming events and also resources, events that we think may be of interest to you if you are into bioinformatics. As well, we have a VanBug calendar, which will have all of our upcoming events. And we have Instagram, Twitter, and YouTube where we do regular promos. On YouTube as well, as I mentioned, we do upload recordings of the talk. So if there's anything that you missed or if you weren't able to attend one of our seminars, feel free to watch those. And lastly, you have the opportunity to suggest a speaker for next season. It's under that same contact page. It's just a little form, and we will try our best to contact them and make it happen. We are also open to international speakers speaking on Zoom. So if you have anybody in mind, please do fill out this form. So lastly, we'd like to thank our sponsors. VanBug is sponsored by the Graduate Studies in Bioinformatics program at UBC and SFU, Genome BC, SFU Big Data Hub, UBC Michael Smith Laboratories, Langara College, Stem Cell Technologies, Vancouver Prostate Center, and we are also affiliated with ISCB and our sister groups Monbug and Torbug. So check them out if you're in those cities. So a bit of housekeeping for our Zoom audience. If you have a question for any of the speakers, please ask your questions in the chat and we'll have a VanBug volunteer voice it out for you. Now on to our speaker bios. So our first introductory student talk will be from Nicole Nutza, who is a PhD candidate in bioinformatics at the Holt Lab at UBC. Their talk is titled The Identification of Novel DNA Sequence Motifs that Modulate Gene Transcription in T Cells. Next, we'll have our featured talk by Dr. Inanc Birol, actually my PI, So Dr. Birol is a distinguished scientist at Canada's Michael Smith Genome Sciences Centre. He's also a professor at the UBC Department of Medical Genetics, an associate member of the Departments of Computer Science and Pathology and Laboratory Medicine at UBC, and an associate scientist at the BC Centre for Disease Control. Previously, he's held research faculty and associate positions at SFU, the Illinois Institute of Technology, and CERN, the latter being a knowledge center for understanding the universe and home to the largest particle physics laboratory in the world. To date, Dr. Birol's team has accelerated research with over 140 publications, which have practical applications in both ecosystems and human biology, including medical genomics. As of 2018, he's listed amongst the top 1% cited scientists in the world by Clarivate Analytics in the cross-field category. Currently, Dr. Birol's research interests include the analysis of data from modern sequencing instruments to study genomes and transcriptomes of model species and humans. He directs the Birol Lab at the GSC, which develops bioinformatics tools for de novo sequence assembly, sequence mapping, and downstream data analysis and visualization. He also directs a wet lab at the BC CDC to study antimicrobial resistance and to develop novel strategies as alternatives to conventional antibiotics. And the talk that he'll be giving today is about the wet lab, kind of. It's titled The Battle Against Antimicrobial Resistance from Academic Innovation to Real World Impact. So that's it for my spiel. Nicole, if you want to share your screen, you can begin your talk.
1 second. Okay, can you see the slides?
Yes.
Okay. So my name is Nicole Knutza. I'm a PhD candidate in Rob Holt's lab. I'm going to be talking about the work that I've been doing for my thesis, focusing on identifying motifs that modulate transcription in T cells. So in the Holt lab, we are focused on developing CAR T therapy for the treatment of cancer, which is a form of gene therapy. And at the moment, many individuals who receive CAR T therapy are receiving a therapeutic product that had been manufactured ex vivo, so outside of the body, to express a therapeutic gene. Essentially, this manufacturing process requires isolating a cell type of interest and using gene transfer vectors to deliver the therapeutic gene along with a regulatory promoter to these cells. And these genetically modified cells are then reinfused back into the patient. And while ex vivo manufacturing allows us to ensure only the relevant cell types express a therapeutic gene, it's extremely expensive and complex, and it's a major limitation of CAR T therapy. So due to these limitations, there's a lot of interest in trying to transition towards in vivo gene therapies. This is where cells are genetically modified directly within an individual, rather than having to perform ex vivo manufacturing. But at the moment, we can't actually administer CAR T therapy in vivo due to safety reasons. This is because the gene transfer vector that is used to deliver the therapeutic gene and the promoter that is used to control its expression are both non-specific. Essentially, this means that any cell type can be genetically modified and express the therapeutic gene, which can actually be detrimental to the patient. So in order to get around this, one of the things we can do is create synthetic regulatory elements for a cell type of interest. Essentially, what we would do is identify the DNA sequences that are associated with a gene expression pattern of interest, and then we use those sequences to create synthetic regulatory elements to control the gene. The main advantage of these elements is that they're significantly reduced in size, so they can fit inside the gene transfer vectors that are used in gene therapies. This is typically not the case when you're using regulatory elements that are found in the genome endogenously. So my project is going to be focusing on trying to identify these sequences and create promoters for T cells. So the two main aims of my project were to create a computational framework to identify DNA sequence motifs associated with T cell specific gene expression. We then also integrated synthetic and experimental biology to try and evaluate the activity of these motifs. So in order to understand gene regulation, you have to integrate a lot of different types of genomic data. The first step for us involved using RNA-seq data for a variety of different cell types, and we identified genes that are expressed in a T cell specific manner. And we also came up with a list of genes that are expressed in T cells, but in a non-specific manner. After that, we use DNA-seq data to identify all the regulatory elements in T cells. So these typically correspond to promoters as well as enhancers, which regulate the expression of their target genes from very far distances. In order to identify the genes that these enhancers regulate, we then use chromosomal interaction data, also known as Hi-C or Hi-CHIP. After we have identified our genes of interest and all of their potential regulatory regions, the next thing we needed to do was to look inside these regions to see if there were motifs associated with our T cell specific genes. In particular, we also know that we had to focus on combinations of motifs because we know that cell type specific gene expression is not primarily regulated by a single transcription factor. And in addition to this, we also made sure that we were focusing on identifying novel motifs since these are typically overlooked in gene regulation studies. After we identify different types of motifs in our regulatory sequences, we need to find out which ones were associated with T cell specific gene expression. So essentially what we do is for our T cell specific genes, we count how many of them have a pair of motifs in their regulatory elements. And then we do the exact same thing for 10,000 sets of non-specific genes. Essentially, this just allows you to find pairs of motifs that are more commonly found in regulatory elements for T cell specific genes. So these are the results from that first analysis. The top plot is showing you how many pairs of motifs we found that were enriched in our enhancer regulatory regions and then our promoters for our T cell specific genes and then genes that are expressed in a non-specific manner. The one thing you'll note from this plot is that a large number of pairs of motifs were identified in the enhancer elements. And you'll also see that there's a large number that were identified for the T cell specific genes and very few that were enriched for the genes expressed in a non-specific manner. We also saw the exact same trend when we were looking at pairs of motifs that comprised novel motifs alongside a transcription factor binding site. So overall, these results showed us that there may be interactions occurring between transcription factors and novel motifs that have not been characterized before, and that these are primarily located in enhancer elements. The next part of my project, which is probably the most exciting, was trying to functionally validate some of the novel motifs for regulatory activity. So we chose 18 candidate motifs to evaluate. There is 9 transcription factor binding sites and 9 novel motifs. And we used a multiple parallel reporter assay in order to do this. Essentially, what we did is we created a library of around 60,000 synthetic regulatory elements. And we made sure that we were accounting for variables that we know are able to influence the ability of a transcription factor to regulate gene expression. So this includes different combinations, orientations, copy numbers, and positions. Essentially, our library allowed us to look at all of these factors for all 18 candidate sequences that we wanted to evaluate. One of the first things that we looked at from our assay was whether or not our novel motifs were able to affect gene transcription in Jurkat cells. So that's a T cell cell line. This plot is showing you all the motifs that we tested on the x-axis and then the activity score. Essentially, the activity score, if it has a positive value, that motif is enhancing transcription. If it's a negative value, it's repressing transcription. And any of the bar plots that are shown as a solid blue color were able to affect transcription in a significant way. So overall, 5 of the 9 candidate motifs that we were testing for regulatory activity were able to affect gene transcription in Jurkat cells. And one of the interesting things we noted is that these effects were often restricted to a particular orientation. So for example, 2802, when it's in the reverse orientation, is able to significantly repress transcription, but when you put it in its forward orientation, it loses this ability. Another thing that we noted, but I haven't shown in this plot, is if you increase the number of copies of a given motif in a synthetic regulatory element, you can essentially increase its activity, whether or not it's activating or repressing. The next thing we wanted to do was to see whether or not combinations of motifs could also alter transcription in T cells. So this heat map is showing the activity scores for pairs of motifs in Jurkat cells. So for example, this top row here is showing that BADF June, when you group it together with any of these motifs, it is enhancing transcription. But when you pair it with motifs that are located in this quadrant, it's repressing transcription. So essentially, this means that pairing motifs with different motifs can have alternative effects in your cell type of interest. In total, we identified 62 pairs that had a significant effect on transcription. And we also noted that, again, the order a pair of motifs appear in a synthetic regulatory element can also change its effect on transcription. So overall, we showed that regulatory elements for T-cell specific genes are enriched for pairs of transcription factor binding sites, as well as novel motifs that have not been characterized before. And these are mostly located in enhancer regulatory elements. We also showed that a subset of the novel motifs we identified were able to affect transcription in Jurkats, and similar to transcription factor binding sites, they're affected by factors such as copy number, orientation, and position. And overall, we believe that this study really highlights the need to expand our search to include novel motifs when we're studying gene regulation, since they're typically overlooked in previous studies. So I just wanted to thank the members of my Holt lab and members at the Canada's Michael Smith Genome Sciences Center that were integral for the wet lab and experimental validation aspects of my project.
Thank you, Nicole. We have some time for questions. For those of you on Zoom, remember to ask your questions in the chat. For those of you in person, you can press the button on your desk and it will add you to like a queue. So when it's your turn to talk, your mic will automatically turn on. So.
Hi, thank you so much, Nicole, for an awesome presentation. I'm curious how this part of the project with the regulatory systems, when they occur, how they play into the rest of the CAR T therapy workflow. Thank you.
Great question. So I believe if I'm understanding correctly, the way that this would translate in the therapeutic setting is essentially we would have to try and find some of the synthetic regulatory elements that we have created and validate them in other cell types to make sure that they're only functioning in T cells. And essentially what we would then do is replace the promoter that's currently used in CAR T therapy with this synthetic regulatory element, and then of course have to do more in vivo tests to make sure that it is truly acting in a T cell specific manner to control the expression of the gene used in CAR T therapy.
So, yeah, great talk, Nicole. So, very interesting work, and I was wondering, your results about distal motifs having a more significant effect on the regulation. Were you expecting that?
Yeah, so it's been shown in studies before that you can achieve some level of cell type specificity from just a promoter or the core promoter, but predominantly cell type specific gene expression is due to motifs within the distal regulatory elements and multiple elements working together to control gene expression. So I wasn't surprised by that result.
Fascinating, because at least to me, it sounds quite counterintuitive. I would expect the cis motifs to be here more effective in controlling.
Yeah, yeah. So I guess like I said, the promoter region is only going to give you a certain level of specificity, or at least a certain degree of expression, but it's really the enhancer elements or even the repressor elements that are going to be responsible for like fine-tuning that expression.
Mm-hmm. Yeah, very interesting. Thank you.
Thanks.
Great. Thank you so much, Nicole, for your talk.
Thank you.
It was super cool, very exciting work, and I'm excited to see where it goes. Yeah, now I guess we'll have Inanc come up.
All right, how about now? All right, good. So thank you very much, everyone, for coming. It's good to see some old faces in the audience as well as new ones. The talk is going to be very informal, so feel free to interrupt at any time if you have any questions. And I will start by doing a disclosure. I am a founder and the chief scientific officer of a company that is developing antimicrobial peptides as alternatives to antibiotics, and that is going to be the topic of my presentation today. And I have active conflict of interest management plans with UBC and my employer, BC Cancer. And I also would like to acknowledge that I work and live on the unceded land of the Coast Salish peoples, including Tsleil-Waututh, Squamish, Stolo, and Musqueam. All right, so our topic today is antimicrobial resistance. All right, so many of you may already heard of the term, and it's sometimes flaring up in the news. Sometimes it's dwindling down, but it is a growing problem. All right, so by 2050, it is expected to be the #1 killer. All right, so it is going to surpass cancer deaths by that time, and reach about 10 million deaths per year. It is already a big problem. So we already have over 1 million people dying as a result of antimicrobial resistance, which means the antibiotics that we are taking are not curing the patients. And it is not only a health problem, it's also an economic one, that it is going to result in about $100 trillion loss in world GDP. So $100 trillion. All right, or maybe I'll stay over here. Sure, yeah, I can stay here. So, I mean, I'm not good in numbers when there is a dollar sign in the front. To me, the way to conceptualize $100 trillion, which is a huge figure, is it's equivalent to 2 Canadian economies. All right. So if we don't do anything about antimicrobial resistance, that is going to be the economic impact. All right. So how would antimicrobial resistance develop? It is very easy. It's a natural phenomenon, but we also give a helping hand. That is, if we take drugs, antibiotics, we kill the ones that are susceptible to the drug, and the ones that survive would multiply and will take over. All right. Not only that, Bacteria are a very friendly bunch, all right? And they have a peculiar sexual life. In case you didn't know, they exchange DNA with each other, all right? So that exchange of DNA can transfer the resistance genes not only across the same species, but across species as well, all right? So they are quite interesting that way. Causes of antibiotics, of course, we talked about the use of antibiotics as one. Overprescription, yes. If we have in some parts of the world where there are no restrictions on access to antibiotics, people take it for no good reason, and that's the patient's not using antibiotics properly. All right, so let's say for flu and for COVID and things like that. We should have learned by now that antibiotics do not cure viral diseases, all right? And it may come as a surprise to you that antibiotics were being used in farming much more than in human health applications. And that to some estimates before the use of antibiotics in farms were curbed, it was like a tenfold more in antibiotics in agricultural use. And there is put in infection control in hospitals. Have you ever heard of hospital bugs? Yeah, I see some nods. So hospital bugs is a thing. You go healthy in a hospital, and then you catch that nasty bug, and then you are done. And of course, lack of hygiene and poor sanitation are excellent environments for the spread of antimicrobial resistance. And to add the salt to the injury, that there is a lack of new antibiotics in development. All right, so it has been a chronic problem that is being addressed more recently. So this slide is already 2 or 3 years old now, but there are new drugs coming online, so that's encouraging. But it is a long and arduous process to develop the new antibiotics, and the big pharmaceutical companies are not very friendly in developing novel antibiotics. They like to recycle the older ones with new properties for various reasons we can discuss. All right, the solutions we are proposing, and not only us, but many labs working in the field, is through antimicrobial peptides. Antimicrobial peptides are everywhere. So all living organisms use antimicrobial peptides to fend off against infectious diseases, all right? And they are structurally and functionally different from conventional small molecule antibiotics, all right? So what does that mean? That means we have a diverse selection to pick from to adjust the spectrum of efficacy of those antimicrobial peptides, or AMPs in short. So they can be gram-positive effective, gram-negative, or both, and they can even be strand-specific in their action. And they have desirable pharmacokinetic and pharmacodynamic properties. They usually interact with the cell wall, and they do not cause antimicrobial resistance as much or fast as conventional antibiotics, all right? That's why emergence of resistance to antimicrobial peptides happens less frequently. Now, did you know that we are not the only animals that use antibiotics? So there are some birds who are actually using antibiotics as a remedy for diseases. Like this crow, for example. This is a picture taken in 2021 by Tony Austin in Swan Lake Nature Sanctuary in BC. All right. So what this fellow is doing is it's inviting some ants on its body. All right. The hypothesis is that the ant bites secrete some antimicrobial peptides. And when the birds get skin infections, they invite those ants and get some antibiotic treatment. Of course, after ants do their business, they are also food, right? So the bird would eat those ants as well. So the birds are not only getting topical antibiotics, but also an oral one. All right. Now, use of antimicrobial peptides in folk medicine or folk applications is not new. Like, for example, in some parts of Finland and Russia, they have been using antimicrobial peptides as a preservative for milk. All right. So they would have this jug of milk, and they would drop in a frog that would prevent the milk from going sour. All right, so they would preserve the milk that way, and they would drink the milk. I don't know what they do with the frog. but the thinking again is the back skin of the animal is secreting some antimicrobial peptides, and that is killing the bacteria that would spoil the milk. All right. I don't know if they still do that, but it was a common practice until a wide availability of refrigeration. All right. Now, okay, so ants do it, frogs do it, all animals, all organisms do it. They secrete antimicrobial peptides. All right. So if you think about it, if they produce these antimicrobial peptides, they must be encoded in their DNA, right? The conventional way of discovering antimicrobial peptides is you take an environmental sample, like you put an ant in a blender, or take some skin swab from a frog, and then you try to find you try to determine if it is demonstrating any antimicrobial properties, and if so, then you try to figure out, you try to isolate which peptide is responsible from it. All right, that's a very costly, that's a very arduous process. Instead, having the knowledge that these are actually coming from the DNA, if you have the DNA sequence, it must be transcribed into RNA. It must be translated into a protein, and post-processing should give you the AMP, right? Well, if we have the DNA sequence, why don't we start from there? So we can do some sequencing either through the from DNA or RNA, all right? And then we can assemble them, then we can annotate them, and then we can in silico translate them, and do some processing as well along the way. And then the question becomes, is this an AMP or not? For that, we built an artificial intelligence tool to answer that question for us. Now, this is how science works. All right, you start with some premise and assume that all these steps work perfectly, and it reminds me of this Rube Goldberg cartoons. I don't know like if it is still popular, but Rube Goldberg made a living out of these cartoons, building these fantastic processes, right? So in this case, if you want an automatic scratcher, all right? The way to do that is you light a lamp. The curtain catches fire. The fire department comes in, squirts the water. There's a dwarf, why a dwarf? Who knows? Sitting there, reaches out for the umbrella, pulls the string, the ball drops, the hammer it crashes the glass there. Baby pup wakes up. Mama dog starts rocking the cradle, and hence you have that automatic back scratcher. All right. This is very much how science works. All right. So we assume that all these steps, from sampling, from extraction, sequencing, assembly, annotation, prediction, they have to work perfectly, all right? Or almost perfectly, as much as possible. So how do you know that it works? How do we know that we have the back scratcher? Of course, we here being scientists, we don't stop there. We have to validate things, all right? So we validate that this back scratcher works, and that's what we do. All right. So in an experiment, you take the ant, you sample some tissue from the ant. You apply some lysis. You lyse the cells. You select for the RNA. You reverse transcriptase. And then you do the sequencing paradigm, the tag sequencing. You do some bioinformatics magic, and then you feed it into an AI, and then the AI gives you that AMP. All right. Okay. So why do we do this? We employ an AI or a machine learning model because if we look at the precursor genes for antimicrobial peptides, they can be modeled, or you can apply an analogy to a rocket-propelled grenade. All right. So the rocket-propelled grenade is the precursor protein product. All right. So it has a rocket, that is the signal part. It has a propeller, that's the pre-pro piece. The signal tells that it should go out the cell. Pre-pro takes it to wherever it needs to go, and then you cleave it off, and then you have the live grenade, the AMP. All right. Now, the AMP part has a low sequence conservation. So you won't be able to look at a sequence and say it's an AMP by a homology search. All right. Now, sequence conservation is moderate on the pre-pro piece and high on the signal part, but there are so many proteins with that signal part that you won't know which ones actually encode for an AMP or attached to an AMP. All right, so enter machine learning. So we have, throughout the years, developed a number of machine learning tools that I'm briefly going to talk about, Amplify being one of them. And for the 501 students in the audience, we're going to dive deeper into Amplify next week. Then we have, so Amplify, what Amplify does is it looks at a mature peptide sequence, and it tells whether or not it's an AMP. So it has a yes or no answer at the end. Okay, so a peptide can be antimicrobial, so it can kill the bacteria. That's good, right? But we don't want it to kill the host, right? So you want to take the antibiotics, you want the bacteria to die, but you hope not to die. So if something is toxic, you want to get rid of that in the development pipeline as soon as possible, and that's why we developed the tool called Tamper. So it's a toxicity predictor. Rampage is a data mining pipeline to automate this process, and Amped Up is another tool for de novo AMP designing. And the student names are written there, so all those students graduated after they published these work. All right, so how would antimicrobial peptide prediction work? So it's an AI, it's a deep learning model, Tamper, Amplify is, and the essence of it is trying to extract some features automatically. All right. So you have seen, you have watched your share of cat and dog videos on YouTube, I'm assuming. Yes, I see some nods. All right, guilty as charged. So when you search for a cat video or a dog video, how would YouTube know? All right. So looking at a picture like this, would we have any difficulty of figuring out which one is a cat, which one is a dog? No, right? The one on the right is a cat, the one on the left is a dog, right? We all agree on that. How do we know that? How do we make that judgment? All right. Okay, so our task is to classify dogs from cats, all right? So distinguish dogs from cats. All right, one way to do that, I mean, looking at these two beautiful pictures, if the ears are down, it's a dog. If the ears are up, it's a cat. Fair enough? Okay, so we'll write a code, we'll write an algorithm to say if the ears are up, it's a cat. All right. Okay, dog. Well, 5 and 6, we have a problem, right? So ears are up, and they're definitely not a cat, right? Does anybody think that these are cats? No. All right. So that feature failed. Okay. How about this? If I see the tongue, it's a dog. If the mouth is shut, it's a cat. All right. Okay. So works perfectly. So I wrote some features. I wrote some rules, and it works perfectly. Okay. So 9 through 16. Dog, cat, dog, cat. Dog on 13? Is that a dog? Would you encounter something like that on the street? You see the tongue and think that it's a dog and go for a pet? No, it's a cat. All right. So, all right, it didn't work. We have to apply something else. I mean, as humans, we are very good at this game, right? We know a dog when we see a dog, like this, I don't know, 4,000-year-old depiction of an animal. Is it a dog or a cat? It's a dog, right? I mean, it's a funny-looking dog, but it's still a dog, right? And we can go even further back in time. So this little animal lurking at the background, my pointer does not work, but that little animal lurking at the background, commingling with humans, is the dog or a cat? It's a dog, right? Again, a funny-looking one with some artistic freedom, right, in its features, but it's a dog, right? Let's come to our date. An animal drawn by a 4-year-old. Is the dog or a cat? Cat? Who says it's a dog? And who says it's a cat? It's a cat, right? I mean, again, it's a very funny looking cat. It has no features about the cat. Good luck finding some features to teach a computer program to say that it's a cat. But if you show enough pictures of cats and dogs, the programs will pick it up. All right, so we have established that feature engineering is problematic, but we can approach the problem from a deep learning perspective because we have enough data, we have enough samples. So if we show enough numbers of labeled data to a computer program, it should be able to start distinguishing cats from dogs. All right. Still, there's a problem. All right. So if I were to pick any peptide out of, say, the human genome, all right, so encoded by the human genome. Humans have some defensins, antimicrobial peptides, but they are not in the thousands, all right? So we have a signal-to-noise ratio problem. So in other words, if I were to pick a random peptide, is it going to be an AMP? More likely than not, it's not going to be an AMP, all right? So I don't know if there are any Lemony Snicket fans in the audience, but it was a big thing when my kids were younger. So they were a big fan of the a series of unfortunate events series. For those who have read the books or watched the series on Netflix, the austere Academy version has a plot. The villain, Count Olaf, is troubling the Baudelaire orphans. All right, the protagonist of the story. So he's always trying to kill them. All right. Now the orphans go from place to place to get away from Count Olaf, and in this episode they're at a school called the Austere Academy. All right, and they built a system to automatically detect if Count Olaf walks through the door. All right, here's how they explain it. Oh, where's my cursor? Okay, there it is. I don't have the sound. All right. So anyways, that's an advanced computer system, all right? If Count Olaf walks through the door, they expose it, and the computer says, not Count Olaf, all right? The problem is that it's dubious, all right? Because there's only one Count Olaf, and if the only thing the computer says is not Count Olaf all the time, it will be right almost all the time, except once. All right? So false negative rate is negligible. False positive rate is 0. All right. So the problem we have is if you have a use case where your candidate AMPs are very few in your system, in your test set, and that's what you want to find. You have to train your system to be sensitive about that one Count Olaf. All right. Okay, so we have built this the system, advanced computer system called Amplify. All right. It represents these peptide sequences as one-hot encoded vectors in the input layer. It is passed into a bidirectional long short-term memory layer, and then it is fed into an attention layer, another attention layer, and the output activation layer. And then we have the conventional five-fold cross-validation. And we have trained it for two particular use cases where you have where you have your input peptides to be equally likely to be AMP or not AMP, or less likely to be an AMP than an AMP. All right. Okay, so this is a deep learning model. It has multiple layers. Okay, but when you are trying to justify why you are using the system as opposed to something else, we have to keep in mind Occam's razor. Occam is a 14th century monk from England. All right, and I'm not going to butcher Latin there, but he expressed that in Latin. Plurality should not be posited without necessity. That's what he said. All right. So, looking at these two animations, do you have any idea what I'm showing there? Solar system. Yes. That's right. Earth-centric. So that blue dot in the middle at the left is Earth. The big yellow sphere is Sun, and everything is revolving around Earth. And then the other one, everything is revolving around the Sun. All right. Which one looks simpler? Right? Okay. Yeah, the one on the right. Okay. Now, of course, it does have some simplifications. Namely, in Copernicus's case, everything is in circular orbit. All right, so he kind of oversimplified it. Now the thing is, and it's a lesson to be learned even today. He figured this out, but he was afraid of getting it published. All right. So Copernicus is also a monk from Poland, Prussia at the time. He only agreed to publish it in his deathbed. All right, because he was a part of a religious organization, and religious organizations are not best known for their freedom of expression. All right, so he kept it to himself. He only agreed to publish it in his deathbed with the preamble that it's just a model. All right, it does not have to reflect reality. It's just a model, and it has better properties. It has better like a simpler explanation of what we're observing. All right. Okay, so coming forward to the Amplify model, you have to show that the complexity you are introducing to your system is actually doing something. And in the machine learning world, it's called ablation. You have to do ablation studies, all right? So with the Amplify model, we just work with the BioLSTM layer, included the context attention, included the multi-head scalar dot product attention layer, and then ensembled them. And every time we increased the complexity of the model, the performance of the model improved. All right. So if a simpler model would work, you would stick with that simpler model. All right. And then, of course, as any good bioinformatics method should do, it should compare against whatever else is out there. All right, so that's what we are showing with the comparisons against AMP scanner version 2. All right, and then we also looked at the performance of this approach, and I'm not keeping track of time. I'm assuming somebody will say something about it if I'm going over time. So we also trained 2 versions of this model for a balanced training set and an imbalanced one, and we showed that for the use case in mind, the proper model is working better. All right. And again, another way to benchmark this is through a receiver operator characteristic curve, or a true positive versus false positive rate. And we showed that the amplify model is at the top there, which is the place to be. So, all right. So I will move on to the temper model. All right. The temper model is slightly different from amplify. When we developed amplify, the transformer technology was not invented. Protein folding problem was not solved. All right, so we are going back in time for the amplify model. Right. By the time we developed temper, by the time Hussein developed the temper model, protein folding problem was solved, and transformer technologies were in fashion. So he used language models for proteins from ESM2 to build representations for the sequences in the input. All right, and sent it through bidirectional GRUs. He was coming up with some representations, some embedding representations for the sequences. In parallel, he looked at the predicted structure of these AMPs. He built a graph neural network, and with message passing in that graph neural network, he came up with another embedding representation for the same AMP, so same peptide, I should say. And then he concatenated those, sent it through the self-attention layer, did some further tricks to predict whether a given peptide is toxic or not. All right. Now, sure enough, we also did some ablation studies to justify the complexity of the approach. But something interesting happened when we looked at the performance of temper. Of course, when you compare it to the alternate methods out there, it was performing better. We were hoping so, and we confirmed that using some numerical experiments. But what we did not expect is that the score, the confidence score it was generating for those peptides would have any correlation whatsoever with the level of toxicity of those peptides. All right, so think about it for a minute. We just trained the system on a binary classification problem. All right, but what we also had as a kind of byproduct was that the score it generated correlated loosely, at least, with the level of toxicity of those peptides. Isn't that cool? That's cool, right? That's emergent phenomenon. And actually, we are reaping the benefits of such emergent phenomena through the large language models we love to work with these days. the ChatGPTs and Claudes and others. All right. So this is similar to what we have in the large language models. When you take king, a vector representation, an embedding representation of the word king, all right? And if you subtract man, again, the vector representation of man, and add the vector representation of woman, what do you get? You get queen. You get the vector representation of queen. That's crazy. That's crazy, right? But that's the principle all these large language models work on. So you train a large language foundational model, and this is what happens, right? So there is some justification about making your system complex enough so that there would be some emerging phenomena, all right? So enter TRIUMPH. So the TRIUMPH is a target predictor for AMPs. Berke is working on it. What he's doing is he's inputting a pathogenic genome, a bacterial genome, peptide sequence with some large language modeling, with some embedding technologies. He is distilling it down to a single vector. All right, so that's HPAT-LAT on the top line. It's a vector representation, and then you have the peptide going through a similar pipeline, getting another vector representation, and the question he tries to answer is this vector effective against the other vector? All right, so it's a very complex model, and he is already reporting. So this is kind of a dated slide. He was already reporting more than 80% accuracy, and I don't recall what the latest figure is. Maybe he can blurt it out.
Yeah, around that vicinity, I'd say.
Yeah, still around 80%.
Yeah, accurate. Yeah.
Okay. All right, so the link to that king minus man plus woman equals queen businesses, once you train a system like this, our hope is it is going to go beyond its training domain, and it would be able to predict things like effective peptides against viruses, against fungi, and other cool applications. All right, so that's the bioinformatics part of my talk today. So we started with our humble beginnings in 2019. Our first tool was built on a database called BART. So Bullfrog, transcriptome repository, and we were able to discover 7 AMPs out of that through sequence homology on the signaling part, and we were able to figure out if it might be an AMP. All right, and then we built the Amplify and published in 2022. We discovered 75 more. And then with Rampage, we were on Rampage. So we discovered the 1,000. We amped it up a little bit. We discovered, or designed, I should say, 7,000 more. And with SwissProt mining, once you have those tools, you can apply it to anything, everything under the sun. So we applied it to the SwissProt database to discover 8,000 more AMPs. The project that supported this adventure was, for the most part, a project called PET-DATE. It was a consortium between Genome Sciences Center, BC Cancer, University of Victoria, UBC Center for Disease Control, I should say, UBC and Amphirax. And we were looking at a pipeline from genomics and bioinformatics all the way to the translation, of course, at the core. This was a One Health project, which recognizes that human, animal, and environmental health are all linked. All right, and we have a large group that was working on this project, which we wrapped up this summer. And as usual, Genome Canada projects do not come 100 cents a dollar. They want you to bring in co-funding. The co-funding was provided by Genome BC, MyTax, UVic and UBC through student scholarships, Chicken Farmers of Canada, Egg Farmers of Canada, Canadian Hatching Egg Producers, BC, what is it? Broiler Hatching Egg Commission, and the Amphirax, as well as Investment Agriculture Foundation of BC. So we put together all those money pots and stirred them to develop some antimicrobial peptides for chickens. So the project was mostly hosted by BC Cancer, and we were curing chickens. So it's a funny mix. But like researchers are like cats, all right? So cats and dogs, we're like cats, all right? How do you herd cats? Can you herd cats? Can you? Like you can, if you dangle some food, they will follow, right? So for us, the food is funding. Genome Canada was willing to give us funds to develop antimicrobial peptides for chickens. All right, so that's what we did. All right, so we again developing antimicrobial peptides and showing them in the lab is one thing, and taking them to the field and showing some efficacy is something totally different. So we started 5 minutes. All right, I should wrap up. So we teamed up with the industry in Abbotsford, Rosedown Hatchery and S.J. Ritchie Research Farms to administer our peptides into fertilized eggs 3 days before hatching. And then we follow them through a full production cycle of 35 days. We started with placing 50 birds a pen, distributing controls and treatments in orange and yellow, females and males segregated. And then we followed them for 35 days to see what is happening. And as you would expect from birds migrate. All right, so we developed some protocols to track the migrating birds to put them back in their pens. That's what we did here with that colors in their wings. Anyways, at the end of the day, we want to show whether or not these AMPs are doing anything. So you start with thousands of AMPs, maybe over 10,000 AMPs. You validate some of them in the wet lab. You take some of them into further characterization, and you take some of them, in this case, 5 or 6 of them, to animal experiments. All right, so I will just quickly show that some of them did show statistically significant effect on the survival of these birds. So we are excited about that. And when we look at what is happening in these birds, so this is another experiment series that were conducted in collaboration with Vito Intervac at the University of Saskatchewan and University of Victoria, we look at how these birds are coping with bacterial infection. So these poor animals are injected with avian pathogenic E. coli, and through 7 days, we check to see if the infections are cleared. The numbers are low. That is indicated by those error bars, right? So we don't have statistical power to say, looking at an individual data point to at some statistical confidence that is helping our case. But when you look at multiple doses, as we're doing here, you can see a dose response, and that dose response is statistically significant. So we are, again, happy about it. All right, we recently reported our findings in a bioRxiv paper preprint, which we are submitting for publication. All right, so I will finally acknowledge contributions from people in my lab. So the names highlighted in red directly contributed to the work I presented today. I'm sure I must be missing some along the way. So with that caveat, I thank you for your attention. I'll be happy to entertain any questions. Yes, Vlad.
Hi, sorry, I pushed one. Sorry. So thank you for your great talk. I had a few questions, but I'll start with one. So I saw that for Amplify model, you had two classes, right? One was peptides, one was AMP. And my question is that, like, where does these labels come from? Like, do you have the access to the label for that? Thank you.
Yeah, good question. An excellent one. So antimicrobial peptides are not new, all right? Antimicrobial peptides were actually discovered before penicillin. We knew about their existence from 1920s. Between 1920 and 2019, I guess, is when we developed Amplify. In those 100 years, there were something like 4,000 AMPs discovered and labeled. That was our training set. Positively labeled training set. The negative labels were selected from SwissProt, excluding some certain keywords about the functions of those proteins. But yes, we did have some positive tests to positive cases to train from. Vlad.
Thanks for the great talk. I have two questions. You mentioned in your example with embeddings that you can remove the embedding of the word man from king to get and add word woman to get queen. Do you think you could do something similar given an effective but toxic AMP to remove toxicity? So if you have embedding for toxicity, you try to remove toxicity from the embedding of an effective AMP to produce a new AMP that is just effective but not toxic.
Interesting thought. So yeah, very interesting. But for that, you would need a decoder for the embedding representation, right? Because
yeah, you would need to produce a novel.
You will have to yes. Cast it to a sequence essentially. But it is very interesting.
And another question is, what presumably we already know a bunch of AMPs. What's stopping them from taking over antibiotics right now? Why are they not?
Yeah. So antimicrobial peptides, again, were known for a long time. The reason why they were not developed as antibiotics were primarily here due to two reasons. One is their bioavailability. By that, what I mean is, if you take antimicrobial peptide orally, enzymes in your gut are going to digest it. So you would get good protein. So it'll be good protein supplement. And second, because of that, antimicrobial peptides are usually administered through injections, right? But at least earlier studies with antimicrobial peptides were conducted before established randomized trial protocols, all right? What it did to antimicrobial peptide research is something terrible, because earlier antimicrobial peptides were toxic. So there's one famous example, that's colistin. Colistin is a peptide-based antibiotics, and is a drug of last resort for certain infections, including UTIs, urinary tract infections, and some lung infections as well, especially for cystic fibrosis patients. The thing with colistin, and again, even that is changing. The thing with colistin is it cures you, but it damages your kidneys. So it also kills you, right? So, but that is changing because the way colistin is administered is having a reconsideration. The dosage, the timing, and all those, the formulation. That's why antimicrobial peptides are having a renaissance, if you will. So that there is a stronger interest in antimicrobial peptides, because we kind of exhausted the simple small molecule antibiotics domain. All right. So we are taking a second look at antimicrobial peptides, and I think it is a good field to be in, because there is a developing understanding about why they should work. I mean, if you think about it, saying antimicrobial peptides are toxic is fundamentally wrong, right? Because all living organisms produce antimicrobial peptides. It's about finding the ones that are not going to be toxic for you, right? And again, the issue is you cannot, for example, administer human antimicrobial peptides to humans. Can anybody guess the reason why not? Why we shouldn't do that? Like there's a company in the Netherlands that is developing antimicrobial peptide-based drugs for cattle, using peptides in the milk, in the peptides in the cattle milk, in the cow milk. Why should we not do that? Yes, exactly. So if we cure for a while some infections, the moment those infections gain resistance to antimicrobial peptides that we use, the entire humanity is gone, right? So we are shooting ourselves on the foot. So we should not do that. All right, where did I come to to this topic? Did I answer your question?
Yeah, thanks.
All right. Yeah, man.
How do I disable this one?
Press the button again.
Yeah.
Oh.
So the mics are on a queue system. So actually, whoever pressed their mic next, their mic is on.
Jonathan.
So when it's flashing, your mic's not on.
I have a somewhat rudimentary question. So you mentioned that AMP, because of its mode of action, it takes longer for bacteria to gain, resistance. So what's to say that when we develop AMP en masse, we blast the population with it. It's an arms race, right? Have there been studies that actually like quantify how much longer a like bacteria takes to gain resistance for AMP versus antibiotics? Yeah.
Ali, would you like to answer that question?
Well, it kind of depends on what you're talking about. Sometimes in studies they kind of expose the population of bacteria for consecutive days, usually for 2 weeks. We have done some small virus studies, and in 14 days for some AMP we have seen emergence of resistance, and for our star candidates we haven't seen any changes in it. So I guess there are studies like that, and it's a more of a comparison how well a small antibiotics will work versus how activity will change.
So yes, and of course that's related to his thesis work, right? There is some semi-quantitative answer to that, and that goes to the dose response curves, right? The reason why some antimicrobial peptides have a narrower mutant selection window is because of the pharmacodynamic properties of the peptides. That is, they have a very sharp response between no killing to full killing, right? Whereas for conventional antibiotics, the slope is very shallow, so it leaves some room for mutant selection. So that's reason number one. Reason number two is antimicrobial peptides. Most antimicrobial peptides do not interact directly with the DNA or the RNA of their targets. That's why they don't call mutagenesis. That's why bacteria cannot explore the fitness landscape to figure out what would cope with the antimicrobial peptide. So it's a multifaceted answer, I guess, to your question.
Thank you.
All right.
I'm going to have a quick nitpick, if that's okay, and then go for a more interesting question.
Sure.
The nitpick is that you showed the effectiveness of the prediction with ROC curves instead of PR curves. And for a class imbalance data set, PR curves would be more appropriate. I'm sure you've done those things internally. Does it still perform with a PR curve?
Yes, it does. So it's a very good question. So anytime we look at the performance of any algorithm, there's one Wikipedia page I load and reload, and that's the sensitivity and specificity, which talks about all sorts of metrics like positive predicted value, negative predicted value, false discovery rate, and all that. So you are right. Depending on your problem, depending on the positive and negative data in your set, some metrics, some measures are better than others, more relevant than others. Yes.
Sweet, appreciate it. I work in sexual health, and the doxycycline resistance for gonorrhea is a constant stressor in my field. So thank you for this work. My question is around for the avoidance of resistance, long-term, those kinds of things, if there is any room for some of these prediction models to include multiple drugs at the same time. I know the complexity of it grows a lot, but we saw with HIV, for example, that some of the best ways to counter resistance is to have multiple medications acting at once. And I'm wondering if that's possible for these models.
Right. The short answer is we don't have enough training data for it, right? But but hey, lack of training data, whenever did that stop us from exploring, right? so it's an interesting question. The problems you have to overcome for a strategy like that is perhaps lies in the realm of molecular dynamics, because peptides can interact with each other. That interaction can be synergistic. It can be detrimental, so they may be annihilating each other, the effects of each other. So that combinatorial effects will have to be measured, and the other part of that answer lies in the work of, again, Berke, where he's looking at peptide-to-peptide similarities and their common targets, right? So that kind of inference on a network structure is going to be informative in that direction. But again, it's Rube Goldberg's back scratcher. You have to validate your results.
Makes sense. One last quick question. Are these, they tend to be smaller, right? These AMPs, are they usually disordered proteins, or are they more fixed in terms of functionality, shape, structure?
Right. So I did not introduce it here. If you are in 501, you are going to see some classification about their structures. Most of the ones that we have tested are alpha helix conformations. There are some unstructured ones, and the one that is causing resistance, Ali was mentioning, is an unstructured peptide. But then there are also beta sheet conformations and alpha beta conformations as well. But because these are short, the estimation of their 3D conformations is, I think, less reliable. But then again, I will pull my Copernican card. It has to be a useful model. It does not have to reflect reality. Maybe it does, maybe it doesn't. I don't care. If it is useful, I will use it.
Thank you.
All right. Yeah, man.
Hi. I have two questions. First one is more on the concept of toxicity and effectiveness. Are these different things? Because if an AMP is toxic, is it selectively toxic towards a bacteria and not the host cell somehow?
Right. So that's an important semantics, right? Yeah. Of course, bioactive AMPs should be toxic to something. And we hope that toxicity is selective towards bacteria and not eukaryotic cells. The structure of bacterial cell wall and eukaryotic cell wall are substantially different in charge and complexity. The AMPs are usually effective against bacteria, but not against eukaryotic cells. But having said that, like in the case of colistin, peptide-based drugs can be toxic to eukaryotic cells. And again, going back to the semantics of toxicity, everything is toxic, right? Even water is. If you drink enough water, you will die, right? So it's about the dose. It's about the therapeutic window between efficacy against your target and safety for your own cells. So that window, if that window is large enough, then you have a therapeutic candidate in your hand.
Thank you. And I also was curious about, so we have discovered, as you shared before, like at this point, thousands of different kinds of AMPs. How do you know which AMPs will act against which kind of bacteria is this done through wet lab testing, or I don't know? That's my question.
Yeah, so the data are only becoming detailed enough for machine learning models to be effective in that front. But that's exactly what the thesis of Berkey is about, making that selective targeting.
I see.
Hi, do we have enough time for more questions?
How are we doing in time, Emily? Are we okay?
Okay, thank you. So my question is that, I mean, it's kind of related to the question that was asked. I wanted to know, like, how much we know about the features that make a peptide and AMP. And I wanted to know, like, can we use the models to just generate, like, imagine that we generate like a ton of random amino acid sequences, and then we give it to the model, and we can test and see if the model predicts they are AMP or not. So we kind of like synthesize AMP, like novel AMPs. So how is it like?
Yeah.
Thank you.
So the answer to your first question is no.
Right.
We don't know what makes a peptide sequence an AMP. I mean, as humans, or at least as a naive person that I am, when I look at a peptide sequence, I cannot tell whether or not it is going to be an AMP. Now, I should qualify that answer, because some people who have worked throughout their careers on antimicrobial peptides can look at a sequence and have a pretty good guess of whether or not it is going to be active. I'm not one of those people, right? For your second question, what was your second question?
So like generating random.
Oh, right. Yes, thank you. So obviously, once you have a classifier, you can use it in many interesting ways, right? Now, of course, if you were to generate randomly the space of possible peptides is huge, right? So with every letter,
yeah,
it is multiplied by 20.
Yeah.
So 20 is to the power, say, 15 is a very big number. Mm-hmm. So you cannot exhaust the space there. And by randomly generating those peptides, running into something that is going to be positive in the antimicrobial peptide question is going to be about the false discovery rate of your algorithm. So you cannot go about doing that randomly. Having said that, once you have some understanding of effective peptides, all right, you can mutate them to make them better. Yeah. Right. So that is one strategy that we applied to a certain level of success with the peptides that we have discovered throughout the years. And the second thing we did, and I did not talk about that, but I flashed the name of the tool in my AI slide. We have a tool called Amped Up. All right. So what Amped Up does is it's a recurrent neural network. All right. It's a very simple, like Copernican simple, circular orbit simple neural network. Okay. So it's an RNN. It's a recurrent neural network. You tell it, okay. So my first amino acid is an arginine. What is the next one? 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 When does it stop? Okay, so it stopped. Is that an AMP? Mm-hmm. So it builds AMPs by randomly seeding starting peptides, starting amino acids, right? Mm-hmm. And interestingly enough, it is beating more complex models, right? So it is a Ptolemaic model versus Copernican model. Amped up is our Copernican model. It works better than the more complex ones. So we have generated some 8,000 candidates using amped up. We were not able to validate all of them, but we selected some few from that list to show that yes, its predictions are actually working. Mm-hmm. So they validate in the wet lab.
I'm also thinking that maybe like bias mutation of amino acids, like the bias search could also help to, like, instead of like searching the whole space of all possible peptides.
Yeah. So we did do those mutations, and we kind of biased it with our preconceptions, like higher the charge, the better it is kind of deal. So we improved the charge of those peptides to show that their bioactivity was improving.
Yeah, thank you.
All right. So stop here. All right, I'll stop here, but I'll be happy to answer any questions during the mingling. All right.
Thank you so much to Dr. Birol. There's pizza and drinks, I think, probably outside. Please feel free to go grab it. You can't eat in here, so just bring it out into the atrium and you guys can chit chat there.